Okay, so very good morning and welcome to Excelsior Academy. So in this lecture today we are going to study about active transport. In the previous lectures till now we were learning the passive transport where only protein as a carrier was needed but no energy was required. In today's lecture we are going to start with the active transport where energy is required in the form of ATP. Okay, so active transport, it is the movement of molecules through the biological membrane from lower concentration to higher concentration, and that's why we need energy in order to carry out. So this is, remember, it's from the lower concentration to higher, that is against the concentration, against the gradient concentration. Okay, concentration gradient. Okay, and that's why we need ATP hydrolysis. So here what you need to understand is that, so we know that ATP has got 3-phosphate, right? That's why it is called triphosphate. Now the bond between these are called anhydride bonds, anhydride phospho bonds. Okay, and they carry lot of amount of energy. Now tell me, which bond will give maximum energy? Breakage over here or over here? Which one will give? Who can tell me the answer? If I make a break over here or here, which will give me the maximum energy? So is it the formation of AMP plus 2pi or ADP plus pi which gives me the maximum energy? No idea? Anyone? Sandhya? Anjan?
ADP plus PI, ma'am.
ADP
ADP
plus pi. Okay.
PI.
Actually, the answer is AMP, because what happens is when AMP is formed, you get 2pi. So the breakage over here will give you the maximum energy. Plus, when this pi bond is there, when this is also broken, I get again the double amount of energy. Okay. So what is happening? 2 times the bonds are broken, and that's why I get the maximum energy. It doesn't stay as pi. It doesn't stay as pi. Again, this bond is broken and it becomes total 2pi. And hence I get the maximum energy when there is a AMP formation. Okay, I hope this is clear. Now, so it's a, okay, sorry, the binding and ATP hydrolysis brings the conformational change. Okay, so what is happening is, like, when there is a ligand binding and the ATP hydrolysis takes place, that is ADP plus pi formation takes place, then there is a conformational change of what? The transporters. So initially it is closed, and when there is a binding, that binding induce ATP binding. Now after ATP has been bound, it undergoes a hydrolysis. Now hydrolysis means release of energy is there. That energy is sufficient for the conformational change. So if my transporter was closed, it will get open up. Okay, and it will allow the passage of molecules from one side to the other side. Okay, so it is a vital phenomena for maintaining the balance of ions across the membranes by concentrating the metabolites in a certain organ or cellular compartments, and also by exporting the waste from the cells. So you will see that, like as I mentioned yesterday also, that sometimes bacteria have got the pumps or the transporters in order to expel out the drug molecules. Okay. Can someone just mute themselves? Someone is, okay. Okay. So what is happening is, so sometimes bacteria, so when you give the antibiotics, so these antibiotics are supposed to go inside the bacterial cell, and then they should inhibit the function of bacteria, hence killing the bacteria. But most of the time what happens is the bacteria is resistant. What do you mean by being resistant? It means it has got certain genes which are responsible for the transporter protein synthesis, which results in the expelling out of the drug. It means what? They take the drug, but they have the transport molecules also by which they actually throw out. So it's like throwing the waste from their system. Okay. So this is how the bacteria is able to survive in most of the cases. Okay, so this is how your concentration is present in these cells. So what you have to remember is that, see, Na plus is 15 millimolar, outside it is high. In few references, it is also given as 150 millimolar, saying that it is 10 times the concentration outside. Then you have got K plus inside, maximum, chloride ions maximum, and calcium ions maximum. So what you have to remember is, out of these 4 ions, only Na plus is present in minimum amount. Okay, whereas in the case of extracellular, it is the other way around. I'm sorry, even the chloride also. So NaCl, Na plus and Cl minus. So they are present maximum in the extracellular space. Now this is why it talks about your osmolarity. Okay. So these two are responsible for maintaining the osmolarity of your blood plasma level. Okay. So the transmembrane transporters are associated with energetically favorable transportation, and they are called ATPases. So what are ATPases actually? ATPases, they are actually the, can I call them as enzyme? Can I call them as enzyme or simply protein? Or simply protein? Can I call them as enzymes? Anyone? Bhavika? No idea? Actually, they are the enzymes only. Okay, they are enzymes only. Of course, all enzymes are proteins. They are also enzymes because ATPases are also responsible for synthesis of ATP. Okay, so this also we'll study. Don't worry. So these can be classified into 3 classes based on the structure and function. So what are those? We have got the P-type, F-type and V-type ATPases, of which today we are going to study the P-type ATPases. Okay. Now, what are the examples of P-type ATPases? So what we have to understand over here is that this class of ATPases have got similarity in their amino acid sequence and hence in the structures. Okay. So I'll just write it down. It has got the similarity in their amino acid sequence as well as in the structure. Okay, so if you understand one particular class of P-type ATPases, the rest also it's easy to understand. So let's talk about the P-type ATPases. Now, why it is called P-type? Because these class of ATPases can be blocked by your phosphate analog called vanadate. Okay, so clear? So why it is called P-type ATPases is because, first of all, what you have to understand is that the phosphorylation, that is ATP hydrolysis, when it takes place, the phosphate group is attached to the aspartic acid. Okay, so it gets phosphorylated. Now this phosphorylation leads into the conformational change of the transporter. Okay, so where it is getting phosphorylated? At the aspartic acid residue, amino acid residue. This you have to remember. Now, when this gets phosphorylated, if I have to block this entire ATPase, then there is a blocking analog called vanadate. Okay, the structure is similar to phosphate and hence it is able to block this particular ATPases. Clear? Okay, so let's move on to the next. Okay, so P-class pumps, they are also called pumps or otherwise P-class ATPases. Now, so they are made up of 2 large identical catalytic alpha subunits, each carrying the ATP binding site. Okay, one of them get phosphorylated at aspartic residues during the transport, and they also contain 2 smaller beta subunits concerned with the regulatory functions. So how can we write its structure? It is alpha 2 and beta 2. because they have got the two large identical catalytic alpha subunits and also the beta subunits. The function of beta subunit is to regulate the function of ATPases, where on the alpha subunits you have got the ATP binding site. Now, all P-type transporters have similarities in amino acid sequence and especially near the aspartic residues which undergoes phosphorylation. So this is important to remember. Now this can be inhibited by phosphate analog called vanadate. These are your 1 marks question type topics, okay? So if you see the structure over here of vanadate, it is similar to your phosphate, right? So even that you even phosphate you make like this only. So you have got PO4 3 minus. Okay. Now, bacteria uses P-type ATPases to pump out toxic heavy metals such as cadmium and copper. So this is what we were discussing. So sometimes not only the drugs, but the heavy metals, because bacteria lives everywhere. They are present everywhere ubiquitously. So sometimes if there are heavy metals which is affecting their cellular process, they have got these P-class pumps, by which they are able to pump out these heavy metals. So if you see, there are, you must have heard about the bioremediation also, wherein which we actually, you know, bioremediation or bioleaching, sorry, it's called the bioleaching. So wherein which what you do is you actually spread most of your bacteria over there. It's one of the application of using such bacteria. So these bacteria, what they do, so in those areas where you're not able to harvest the heavy metals, okay, so what they do, they make use of these bacteria, and these bacteria start living on these heavy metals, and since it cannot be used by them, so what they do, they start accumulating, and one find, like after once the accumulation becomes more beyond the cellular process to accommodate, they start pumping out. So what happens, so entire area you can collect it out, you can just kill the bacteria and you can take out the heavy metals. So this is how you can actually do the mining of the ores also, where you know heavy metals can be extracted easily with the help of these bacteria. So one kind of application is also there. So yes, so let's move on further. Now, so now in this case, in the case of P-type classes or P-type pumps, we are going to study about the two examples. So first one is example, the first example is calcium ATPase. Okay, now in this case, in this case, you transfer calcium once a muscle has been contracted. Now there are two types of two types are there. So you have got two types are there, PMCA and SERCA. So what are those? Plasma membrane calcium ATPase and sarcoplasmic reticulum calcium ATPase. So this you mainly see in the case of muscles. Now, whenever calciums are present in the intracellular, concentration is high. How it has been regulated? It is being done with the help of the protein called calmodulin. Okay, so it is regulated by calmodulin. It is important. So it is a cytosolic calcium binding protein. Calmodulin triggers the export of calcium 2+ ATPases to maintain the low level of calcium, that is the resting cell characteristics. As I said, in the case of intracellular, what are the ions which needs to be in less? That is Na plus and chloride minus, right? Whereas the level of K plus and calcium 2+ will be high at the intracellular. Okay, so whatever the value is there, they need to be maintained, and to maintain that we have got the protein called calmodulin. Okay, so whenever we are going to study about the cell signaling also, there also this calmodulin is playing a very important role, because all these calciums needs to be regulated, and it is the calcium binding protein. Is it clear? So there are two types are there, plasma membrane and sarcoplasmic reticulum. Okay, so these two are there. Now we are going to study their structure and their function. Okay, so see the structure over here. So how many transmembrane alpha helices are there? How many are there? So we have got around 10 alpha helices.
10.
Yes, very good. So we have got 10. Now, can you see that these dots are there, red color dots, which is said that it is the calcium binding residue. Now, who can tell me why we have got 4 binding sites? So I can give you one hint that at a time, 2 calcium ions binds. So why do we have four binding sites over here? Can you give me the reason? Ranjan, any idea? Like, why do we have 4 sites over there when only 2 calcium ions are binding? Okay. Anyone else? See, what you have to understand over here is, suppose this is your channel, okay? So your channel is getting open up over here. So it will bind here and here. 2 calciums are binding. Now, when the ATP comes, it is going to do the hydrolysis. Now, from this binding site, it needs to move this side, correct? See, initially it has been bound towards this side. Now, when this has to be moved from this side to the other side, like from cytosol to outside, it will move from 4th, 5th region to 6th, 8th region. यहाँ से नीचे की तरफ जाएगा. Okay, so from the top it will go towards the down, and then when it gets open up like this, then it is easy for them to cross the membrane. Getting the point? See, your transmembrane is this much big. So first it will bind over here from the cytosol. Okay, then when there is a ATP binding, conformational change will take place from the here to here. It will bind then, and when the channel or when the transporter gets open up, it will just go to the other side. Okay, and that's why we have got the 4 binding sites. Now this is just the schematic view of the transporter. We will also see the other structures. So you can see over here that you have got your N terminal and C terminal at both the same side in the cytosolic region. Okay. Now you have got the energy transduction, phosphorylation of aspartate and ATP binding site. So remember we were talking about the beta subunits. So their function is having these globular domains are there where phosphorylation and energy processing is being done. Okay. So this is how the schematic view looks. We'll go to the other topic. So this is how the mechanism. This is how the mechanism is behind the calcium ATPase. Okay. So I try to get the the colorful pictures with each domain represented differently. So you see that you have got over here the membrane lumen and you have got the cytoplasm. Okay. Now, what you have to remember over here is one important point is that the enzyme is going to be in 2 states. Okay, it is going to be in 2 different conformations. What are those 2 conformations? First conformation is when the enzyme is not phosphorylated. Second is when it is phosphorylated. Okay, so now what you have to see is, see, can you see? We have written here E1 and here it is E2, right? So E1 means there is no phosphorylation happened. E2 when there is a phosphorylation happened. Okay. Now let's see. So what is happening now? So the calcium comes, it goes and binds over there, right? Now that causes your ATP to come and bind to the ATP binding site. So can you see? This is your ATP binding site. Now see, this is your phosphate binding domain. So where at the aspartic residue, the phosphorylation will take place. So you can see that when ATP is bound over there and ATP hydrolysis takes place. So here hydrolysis is taking place. So what happened now? So the phosphate group from the ATP has been bound to the aspartic residue and this has become ADP. Okay. Now, once it becomes ADP, the ATP binding site loses its affinity for ADP. Getting the point? It has got affinity for ATP only. So once it becomes ADP, it loses its affinity. Now, when I say loses its affinity, what does it mean actually? So in the binding site, suppose you have got the 3 interacting partners. So only ATP is able to make the interactions. It's just remember your lock and key model or induced fit model of enzyme and substrate complex, right? Now suppose one of the phosphate has been removed. What happens? So the interaction with the binding site is less and hence it loses its affinity for it. So once it loses its affinity, what is happening? The ADP will get removed from the binding site. Okay, so now what is happening? The ATP has been removed. Now, when this ATP was there, now see the structure over here also. See, it was calcium bound over here. Now, once the ADP was removed, that causes the conformational change. Can you see the conformational change? So initially it was open from this side. From the cytosolic side, it was open. Now, once the ADP was formed and there was a conformational change during its release, you can see that the calcium was taken out. Okay. Now, the thing is, the entire enzyme complex needs to be activated back. That is, it has to be brought back to its original form. So, how it will be? Again, it will undergo the hydrolysis where the phosphate group will be removed and the enzyme will come back to its normal position. Okay. Now, here it is called E2. Here it is called E2. Why? Because here you can see that this was closed. It is open. Right? It was closed over here. It is open. So, these are the two conformations under which a ATP is undergoes. So, everything is clear. Any doubts at any step? See, numbers are given 1, 2, 3, 4, 5, 6. So you can just ask me now if any doubts are there. I can explain once again. So do you want me to explain again? It's quite simple actually, if you see. So here it is given that enzyme with 2 calciums are given. Then ATP is bind. So your equation also becomes like that, right? Now what is happening? The phosphate group has been attached to the enzyme and that's why it becomes E1P calcium because these are attached to the enzyme and then ADP. So what all has been removed? Then the calcium is removed, ADP is removed. So your equation becomes E2P. Why E2? Because there is a conformational change. See the structure over here. It is open from inside. Now here it is open from outside. Now once this is there, finally phosphate group is also removed. So your equation is only E2. Now when then it goes back to its normal condition, that is close again. Nothing is there. It is only E1. Okay. So in future, suppose some chemical reaction is given like E1 plus ATP plus calcium 2 plus twice. So what kind of structure are you expecting? Okay. So that structure is going to be this form. So you should be able to understand when the equation is written like this. So it means in your mind, at the back of your mind, this structure should come. Clear? Now, if suppose ATP hydrolysis has been taking place, it is written ADP, so it will be written as E1P. Guys, is it clear? Should we move on? Now, the book which I have shared with you, the Leninger, right? In that also it is given. So, in case if you're forgetting, you can also study from that. Or you can watch the video again, or you can study from the Leninger. Okay. Is it clear? Is it clear? Should I move on? Okay. Now, next one. So the second example which comes under the P-type ATPases is sodium potassium ATPases or otherwise called pumps. So this is the one which we, mainly listen about this because it is involved in maintaining your membrane potential. Okay. So this is the one which is responsible for maintaining the resting potential also. The resting potential of your membrane. Okay. So again, so it's a tetramer of alpha 2 beta 2 subunits. So if you see that the calcium 2 plus ATPases as well as Na plus K plus ATPases, the structure is almost similar. As already mentioned that P-type classes, they have got similarities in amino acid sequences as well as in the structure. Okay. Now in this case, so in the case of calcium 2 plus ATPases, how many calciums were transported? 2 calciums per ATP molecule. Okay, so in that case it was 2 calcium 2 plus per ATP molecule. So this is the formula in the case of calcium 2 plus ATPases. Now in the case of sodium potassium ATPases, it is 3 Na plus and 2 K plus. 3 Na plus in exchange of 2 K plus per ATP molecule. Okay, so you can see that inside the concentration it is 100 millimolar and Na plus it is 10, whereas outside it is just the opposite. So K plus is going to be less and Na plus is going to be maximum. Now one question over here. If suppose I'm saying that I have used a 20 ATP molecules in this particular ATPase pump, so how many K plus and how many Na plus has been transported? Who can calculate?
40 ATP, ma'am. 40 K plus.
40 K plus. What about Na plus?
60 Na plus.
Yes. So it is going to be 40 K plus and 60 Na plus. Simple formula is that 3 Na plus 2 K plus per ATP molecule. Okay, so very good. So if suppose in exam, if suppose it is given that 150 Na plus is transported, so how many K plus and how many ATP molecules you can calculate, right? So it will be divided by 3 in the case of Na plus. So you get how much? 50. So 50 ATP, so that means it will be 100 K plus. Clear? So in any way around you are able to calculate. Okay, good. Now the function of these Na plus K plus ATPs is to maintain high Na plus outside and high K plus inside. So this is what that figure is all about. Now this is also very important, the inhibitors. Urbane and sorry, this is not a spelling actually. Digitoxin. It is a digitoxin. Okay, so they inhibit the ATPase activity. Now how they're inhibiting we'll study in the upcoming slides. Now again it has got the 2 conformation as I mentioned before in the case of Ca2 plus ATPases. So if it is clear over there, then it becomes very easy in this case also. Okay, so you see that if it is phosphorylated, it is enzyme 2. If it is dephosphorylated, it is enzyme 1, as similar case with the previous one. So it has got the affinity for K plus, high affinity for K plus and low affinity for Na plus. In the case of dephosphorylated, it has high affinity for Na plus and low affinity for K plus. Okay, so what does it mean over here? So when, see over there, it was only Ca2 plus, correct? So over there, the phosphorylated and dephosphorylated was explained in terms of ATP. Time is running out. So it was explained in terms of ATP, whether it has got high affinity for ATP or ADP. In this case, it is explained with respect to K plus and Na plus. Okay. Okay. So as I mentioned, so it has got the 2 conformation. Okay. So the enzyme 2 has got the high affinity for calcium, K plus, and the enzyme 1 has got a high affinity for Na plus. So who can tell me which ions are coming first, or which is going where? Meher, can you guess which ions are binding first from the given condition? Yes, anyone else? See, from here itself, it should be clear that when we are talking about the enzyme 1, that is the dephosphorylated state, when there was no ATP binding and hydrolysis, it means that the first ions which are going to bind are Na plus. Okay, so once the Na plus has been taken in, then the K plus are binding. Okay, so let's see the structure. It will be more clear. Yes. So yes. So now you see that it is given that the exterior Na plus is high, and here the interior, that is intracellular, K plus is high. So this is how you represent. Okay, so high of K plus inside and low outside, and the opposite condition in the case of Na plus. So exterior, it is having high concentration of Na plus, and outside, inside it is less. Now this is your Na plus, K plus, ATPase. So you can see that alpha and beta subunits are there, and it has got ATP binding domain. Now what you can see is, it has been highlighted over here that these are your 3 Na plus binding sites. Okay. Now the first thing what happens is the binding of Na plus and ATP. So what is happening then? So once the Na plus is bound and ATP is there, it undergoes conformational change by adding phosphate group at aspartate or aspartic acid, okay, represented as D nomenclature. So now these Na plus are there, ATP is lost, and you have got the phosphate group. Now what is happening? There is a change. Now the 2K plus will come and bind. This will result in the release of Na plus. So what is happening? Once your 2K plus is bound to the enzyme, it loses affinity for Na plus. So your Na plus has been removed. Okay. Now one more thing, just see over here. So you still have 2K plus and a phosphate group. Okay. Now once the phosphate group has been removed, it undergoes dephosphorylation. This dephosphorylation causes the second conformational change, which results in the release of K plus. Okay. So again, these are all happening in your 6 steps, like your previous calcium 2 plus ATPase. Okay, is it clear? So I'll just explain once again. Okay, few people have joined now. So see over here that the exterior Na plus is, okay, many people are joining now. Okay, so exterior Na plus is high over here and low, whereas K plus, it is high inside and low outside. Okay, then we have got the alpha subunit and beta subunit of the enzyme along with the ATP binding domain. So there you can see that there are 3 binding sites for Na plus and 2 binding sites for K plus. Okay, now first step is that your Na plus and ATP binds. So here and here your Na plus has bound and ATP is bound over here. Now once ATP undergoes hydrolysis, what happens? The phosphate group is attached to the aspartate residue, which brings the conformational change. This conformational change brings about the addition of 2K plus. Now when the 2K plus goes and binds to the enzyme, that results in the release of Na plus, 3 Na plus. Okay. Now if you see the structure over here, it is still having 2K plus and phosphate group. Now the dephosphorylation, that is removal of the phosphate group, brings about the second conformational change. This second conformational change leads to release of K plus. Now once K plus is removed, that is from intracellular to external, when the K plus is removed, what happens? It goes back to its normal condition where the 3 Na plus, 2 K plus and ATP binding sites are open again. Okay, so this all happens in the 6 steps. Very much similar to your calcium 2 plus ATPases. So clear? Is it clear? Yes, Ranjan and Akshita, you guys joined late, that's why asking. Yes? Okay, good. So let's move to the next. Now, due to the transport of 3 Na plus outside and 2 K plus transport inside, a transmembrane potential of -50 to -70 millivolt is created. So from this, you should understand that and remember also, because in the upcoming lectures, we are going to talk about the membrane potential, how the polarized and the depolarized state of the membrane is maintained. So for which,
ప్లీజ్ పోవే మూలకి నాకు చాలా బరుస్తుంది
guys,
ఏం చేయాలి చెప్పు మరి
mute
ఒక
yourself.
2 minutes చేసి తర్వాత
వద్దు, నాకు
వేస్తా,
చాలా
నేను వేస్తా.
బరుస్తుందిగా
నాకు కూడా బరుస్తుందిరా
ప్లీజ్
సృష్టి.
Please mute yourself, please, please. Okay. Okay. So due to the transport of 3 Na plus and 2 K plus, the membrane potential is maintained between -50 to -70. So we remember that resting potential is -70 millivolt, right? So how exactly it is -70, we'll study in the upcoming lectures. Now, Obane is the potent specific inhibitor of the Na plus K plus ATPases, as mentioned before. Now, what happens is it binds to the protein that is open to the extracellular side, locking the locking the 3 Na plus ions, preventing its conformational change. So what is happening? So you have got the structure, right? So we had 3 Na plus binding. So this goes and blocks the binding of these Na plus. So what is happening then? It doesn't allow the binding and it prevents its conformational change. Okay. Now it is used for its medicinal purpose. So this is the plant from which actually the chemical has been isolated. So it is also called the cardiac glycoside. So in small amount, it can be used to treat hypotension and arrhythmias. So you can see that, see, many of these, I'll tell you one example, like in the previous, in the traditional methods and all. So usually when people were not knowing about these plants and herbs, so they used to take in high amount. So what happens when you take in high amount? You actually get heart attack. Okay. So all these heart related problems comes. Now suppose now you know that you have got high BP, but if you take this in small amount, what will happen? Your BP will be normalized. Okay. So slowly the Ayurvedic people and the scientists, doctors, they understood about its importance, like how exactly it functions. So now we know that it actually inhibits the activity of Na plus K plus. So now I know I can maintain the potential of the membrane. So the nerve impulse I can maintain. So now as medicine it has been used. Okay. Before also I have mentioned about the Dhatura and Oxia is there, right? So if you use in high concentration, it increases your high BP, it causes hallucinations. It's like kind of drugs abuse. Okay. Now if you use in small amounts, smaller quantity, like in few micrograms, then it acts as a medicine. So this is about the Obane, and there is one more. Okay. So that is digitoxygenine. Okay, digitoxygenine. So it is the active ingredient of digitalis. So what is digitalis? It is this flower. Can you see? I have seen this in many places as the ornamental plants, but I never knew that this can be also toxic if it is inhaled continuously or digested. Okay, ingested, sorry. So it is the foxglove plant and it is used to treat congestive heart failure and strengthens the heart muscle contraction. So what does it mean? So it is going to increase your heart muscle contractions. Now, if suppose a normal healthy person takes this digitalis, what will happen? It will make more contractions, right? And the beating will be very slow. So what happens? The person will die of the cardiac arrest. Getting the point? In the previous, it is because of the high BP, the person will die. In this case, it will because of the arrest. It won't be able to contract further. Now, digitalis again inhibits the efflux of Na+, raising the intracellular Na+ concentration. Now, this will also affect your heart muscle concentration. Now, this activates your Na+, Ca2+ antiporter. Remember what is antiporter? So, if suppose Na+ comes in, your Ca2+ will go outside. Okay. So, antiporter means in the opposite direction at the same time. So it activates your Na+, calcium 2+ in heart muscles. It increases the influx of calcium 2+, which will strengthen the contraction. So if you're using as a medicine, then you have to use it like, in smaller doses. Okay. Okay. So you have to use it in the smaller doses. So that was all for today's lecture. Now, if any doubts are there, you can ask me. Thank you for listening.